Properties of amino acids. Amino acids have the following physical and chemical properties. One is solids. The most of the naturally occurring amino acids are solid in nature. They are in the form of crystals. The crystals form vary from cylinder needles, that is tyrosine, to thick hexagonal plates like cysteine. Color. The amino acids are colorless. Taste. Amino acids may be tasteless, example tyrosine, or sweet in taste, example alanine and glycine, or bitter in taste, arginine. The sodium salt of glutamic acid, that is sodium glutamate, is valuable as flavoring agent for certain sauce of foods since it enhances flavor. It is manufactured in large quantities for this purpose, usually by hydrolysis of proteins which contains large amount of glutamic acid. Fourth one is solubility. In general, amino acids are readily soluble in water, slightly soluble in alcohol, and insoluble in ether. Tyrosine is only slightly soluble in cold water, but more soluble in hot water. Cysteine is insoluble in cold water and hot water. Proline and hydroxyproline are soluble in alcohol and ether. The amino acids are generally soluble in dilute acids and bases, in which they form the amino acid salts. Tyrosine is only moderately soluble in dilute acids and bases. Cysteine is soluble in solutions of strong mineral acids such as hydrochloric acid, but only slightly soluble in solutions of acetic acids. This is also only slightly soluble in dilute ammonia. Melting points. The amino acids are outstanding among organic compounds in possessing high melting temperature or melting point. In general, melting points are above 200 degrees Celsius. Certain amino acids melt above 300 degrees Celsius. Most of the amino acids undergo decomposition at or near the melting point. Optical activity. All amino acids except glycine are optically active. Optical activity is the property of the molecules by virtue of which they can rotate the plane polarized light. Most of the amino acids rotate the plane polarized light to the left, and such amino acids are called levorotatory amino acids, and they are designated as by symbol minus. Some amino acids rotate the plane polarized light to the right, and they are called as dextrorotatory amino acids. They are designated by the symbol plus. The property of optical rotation is due to the existence of asymmetric carbon atoms in amino acid molecules. As glycine has no asymmetric carbon atom, it cannot rotate the plane polarized light. The specific rotations of some amino acids: L-proline minus 86.2, L-histidine minus 38.5, L-phenylalanine minus 34.5, L-threonine minus 28.5, L-serine minus 7.5, L-alanine plus 1.8, L-glutamic acid plus 12, L-isoleucine plus 12.4, L-arginine plus 12.5, L-lysine plus 13.5. All optically active amino acids of proteins are L-stereoisomer. The optical rotation of an amino acid is different for its cation, anion, and zwitterion. The optical rotation varies according to the pH of the solution, which determines the ionic state of the amino acids. For example, the specific rotation of L-alanine in water at 28 degrees centigrade is plus 2.4. In 6N-hydrochloric acid is plus 14.5. The specific rotation of L-leucine in water at 25 degrees is minus 10.7. In 6N-hydrochloric acid is plus 15.1. Ampholytes or amphoteric nature. Amino acids behave both as weak acids and weak bases because they contain at least one carboxyl group and one amino group. Such compounds are called ampholytes or amphoteric compounds. They are capable of both donating and accepting protons. Next one is zwitterions. Amino acids behave as zwitterion. A zwitterion is a dipolar ion containing negative and positive charges. As amino acids contain both acidic and basic groups, they can react with both alkalis and acids to form salts. In an acid solution, that is low pH, amino acids carry positive charges. and hence they move towards cathode in an electric field. The positive charge is due to the suppression of the ionization of carboxylic group. In an alkaline solution, increasing pH, the amino acids carry negative charges and therefore move towards anode. The negative charge is due to the removal of a proton from the amino group. But at a certain pH, the amino acid molecule is electrically neutral, where the number of positive charges equals to the number of negative charges. This occurs as a result of dissociation of the H plus ion, which passes from the COH to the NH2 group. The amino acids possessing both positive and negative charges are called zwitterions. When amino acid is in the zwitterion state, it will not move two electrodes in an electric field. Hence, they are electrically neutral. The pH at which an amino acid has no tendency to move either or the positive or negative electrode is called isoelectric point or isoelectric pH. At this pH, the amino acid molecules bears a net charge of 0. The isoelectric point is symbolized by Pi. The isoelectric point varies from one amino acid to another. Some isoelectric point of amino acids: aspartic acid 2.77, glutamic acid 3.22, phenylalanine 5.48, tyrosine 5.66, tryptophan 5.89, alanine 6, proline 6.30, histidine 7.59, lysine 9.74, arginine 10.76. When an acid is added to a zwitter amino acids or zwitterion, the amino acid becomes positively charged and it moves to a cathode. The carboxylate ion of, that is COO minus of the zwitterion, amino acid combines with a proton H plus of the acid hydrochloric acid to produce an acid COOH, bearing a net positive charge. Hence, it moves to the cathode. When an alkali is added to the zwitterion, that is zwitterion amino acid, the amino acid becomes negatively charged and it moves to an anode. The NH3 of the zwitterion amino acid liberates a proton H plus. The amino acid becomes an organic salt bearing a negative charge. Formation of esters. Amino acid reacts with alcohol in the presence of hydrochloric acid to form esters. In the reaction, first hydrochloric acid breaks up zwitterion structure to form the amino acid hydrochloride, which then reacts with the alcohol to form ester hydrochloride. Next one is decarboxylation and the formation of amines. When amino acids are heated in the presence of barium hydroxide and CO2 is lost and an amine is formed. Here the carboxyl group is removed and hence the process is called decarboxylation. Next one is amide formation. Amino acid esters form amides when treated with anhydrous or alcoholic ammonia. Acylation of amino acids. Amino acids is acylated or acetylated with acetyl chloride or acetic anhydride. Benzolation of amino acids. Amino acids yield benzoyl derivatives with benzoic acid. For example, glycine reacts with benzoic acid to form benzoyl glycine. In this reaction, the amino group condenses with benzoic acid to form epuric acid. This process occurs in men as process for detoxification of benzoic acid. Methylation of amino acids. The amino groups of amino acids may be methylated by treatment with methyl iodide or dimethyl sulfate in alkaline solution. The final product is a betaine of amino acid and the reaction takes place in stages. Reaction with Sankar's reagent. The amino group of amino acids reacts with Sankar's reagent 1-fluoro-2,4-dinitrobenzene, that is FDNB. In this reaction, the reagent FDNB condenses with the free amino group in cold mild alkaline solution, that is bicarbonate, to Q dinitrophenyl amino acid. With the help of this, Sankar determined the sequence of amino acids in insulin. Reactions of amino acids with with nitrous acid. Amino acid reacts with nitrous acid to form the corresponding hydroxy acid with the liberation of N2. Nin-nitrogen reaction, that is oxidative deamination reaction. The amino group of an amino acid may be easily removed by oxidation. This process is called oxidative deamination. When amino acid is heated with nin-nitrogen, the amino acid is detected the NH3 and keto acid by oxidative deamination. The reaction occurs in two stages. The nin-nitrogen reaction is used for the quantitative estimation of amino acids, peptides and proteins. Edman reaction. Phenyl isothiocyanate reacts with amino group of amino acid under alkaline condition to form phenyl isothiocarbamyl peptides. Carbamino reaction. The free amino group of amino acid can condense with carbon dioxide to form carbamino compounds. This reaction is called Segeford carbamino reaction. Carbamino compounds are formed in the food when the amino acid in hemoglobin react with carbon dioxide. Carbamino compounds of hemoglobin are important in the transport of carbon dioxide. Formation of diketopiperazines. The esters of amino acids condense to form anhydride ring structures containing two amino groups. These substances are called diketopiperazines. Chelation of amino acids with metal ions. A number of heavy metals ions such as copper 2+, copal 2+, magnesium 2+, iron 2+, etc. From chelated complexes with amino acids in which both carboxylic and other groups such as NH2 and SH are involved. For example, copper salt of glycine is formed by heating copper oxide with an aqueous solution of glycine. Formation of fatty acids. When amino acids are heated with hydroiodic acid at 200 degrees centigrade, the amino group is eliminated from these amino acids and they form fatty acids. Formation of salts with alkalis. Amino acids reacts with alkalis to form salts. Reduction reaction. Amino acids can be reduced to the amino alcohols by lithium aluminum hydride. Next one is Darkin-West reaction. When amino acids are heated with acetic anhydride in pyridine solution, these are converted into methyl alpha acetamidoketones. This reaction is often referred to as Darkin-West reaction. Sorensen's formal titration. The method of titrating amino acids with alkalis is termed as Sorensen formal titration. The monoamino monocarboxylic acid, that means amino acids having one NH2 group and one carboxylic group, behave both as an acid and as base. Hence, the carboxyl group of amino acid cannot be accurately titrated in water solution with alkali because it reacts with the basic amino group to form zwitterions that are not decomposed completely at the end point of alkaline indicators, that is phenolphthalein. Sorensen observed that if amino acid solutions are neutralized to phenolphthalein and treated with a large excess of neutralized formaldehyde solution, the mixture becomes acid and can be titrated sharply to phenolphthalein with standard alkali. The amount of alkali required for this titration was found to be correspond to the complete titration of the carboxylic group of amino acids. The reaction of formaldehyde with amino acid in the Sorensen titration is complex. The amino group combines with formaldehyde to form dimethyl amino acid, which is an amino acid formaldehyde complex. In this way, the existence of all amino groups are eliminated, resulting in the acidic solution due to the presence only of the carboxylic groups. This solution can be titrated with standard alkali using phenolphthalein as indicator. The amount of alkali required indicates the amount of carboxylic groups present. Color reactions of amino acids and proteins. First reaction is nin-nitrogen reaction. All amino acids when heated with nin-nitrogen can form complexes pink, purple or blue in color. The color complex is called romance purple. Proline and hydroxyproline will give yellow color with nitrite. Amino acids with amide group, that is glutamine and asparagine, produce a brown color. The nitrite reaction may be adopted for qualitative as well as quantitative estimation of amino acids. It is often used in detection of amino acids in chromatography. Proteins do not give a true color reaction, but N-terminal end amino group of protein will also react with nitrite to produce a blue color. Second one is biuret reaction. Cupric ions in an alkaline medium form a violet color with peptide bond nitrogen. This needs a minimum of 2 peptide bonds, and so individual amino acids and dipeptides will not answer this test. This reaction can be used for quantitative estimation also. The name is derived from the compound biuret, NH2CONH2, a condensation product of 2 urea molecules, which also gives a positive color test. Magnesium and ammonium sulfates interferes with this reaction. Third one is xanthoprotic test. The ring systems in phenylalanine, tyrosine and tryptophan undergo nitration on treatment with concentrated nitric acid. When heated, the end product is yellow in color, which is intensified in strong alkaline medium. This reaction causes the yellow stain in skin by nitric acid. Fourth one is Milan's test. The phenol group of phenylalanine and tyrosine containing proteins, when heated with mercuric sulfate in sulfuric acid and sodium nitrite, or mercuric nitrates in nitric acid, form red colored mercury phenolite. Chloride interferes with this reaction, and so it is not suitable to test for tyrosine in urine sample. Both xanthoprotic and Milan's test are negative for tapioca, that is cassava, which is deficient in phenylalanine and tyrosine. Fifth one is aldehyde test for tryptophan. In the Hopkins-Cole test, tryptophan containing protein is mixed with glyoxylic acid, and the mixture is layered over concentrated sulfuric acid. A violet ring at the interface of liquid shows the presence of indole ring. Formaldehyde and mercuric sulfate is used similarly in aqueous romanzine reaction to get a violet color. Para-dimethyl aminobenzaldehyde and strong hydrochloric acid give dark blue color, that's Ehrlich reaction. Gelatin with limited tryptophan content will not answer this test. Sixth one is Shakakushi test for arginine. Free arginine or arginyl residues in proteins react with alpha-naphthol and alkaline hypobromide to give bright red color. This is due to the guanidinium group. Seventh one is sulfur test for cysteine. When cysteine and cysteine containing proteins are boiled with strong alkali or organic sulfur, splits the forms sodium sulfite, which on addition to lead acetate produce lead sulfide as a black precipitate. Methionine does not answer this test because sulfur is methionine is in the thioester linkage, which is difficult to break. Albumin and keratin will answer sulfur test positively, but casein will give a negative result. Nitroprusside reaction for SH group. Proteins with free sulfhydryl groups give a reddish color with sodium nitroprusside in ammonial solution. Many proteins give a negative reaction in the native state, but when denatured, reaction will be positive, showing the emergence of free SH group. Next, ninth one is false test for histidine or tyrosine. Diisobenzene sulfonic acid react with imidazole group of histidine to form a cherry red colored diazotized product under alkaline conditions. The same reaction or same reagent will give an orange red colored product with phenol group of tyrosine. Now summarize the reactions. Biuret reaction, the observation is violet or purple color, which indicates the presence of 2 peptide linkages. Next one is nitrite reaction. The observation is blue color, which indicates the alpha amino acids. Next one is xanthoprotic reaction that produces orange color. The aromatic amino acids phenylalanine, tyrosine, tryptophan will give the positive result. Next one is Milon's reaction. It produces red color, that is phenolic group, example tyrosine. Oppi's cold reaction, it forms violet color ring, that is indole ring, present in the tryptophan, will give the positive result. Next one is Shaka-Cauchy reaction, that is intense red color, which indicates guanido group, which is present in the amino acid, example arginine. Nitroproside reaction, that gives sulfidal groups. Next one is sulfur test, that is black precipitate, which indicates the sulfidal groups, that is example cysteine. The final one is Folin-Sacaltio test, which forms the blue color, which indicates the presence of phenolic groups.